Rhabdoviruses introduction. Rhabdoviruses are bullet or rod-shaped viruses with a single standard RNA genome. They are included in the family Rhabdoviridae. Rabies viruses belongs to the genera Lysoviruses. Morphology: a bullet shaped 75 into 180 nanometer with one end rounded and the other polar or concave. The outer lipoprotein envelope contains a heme-agglutinating peplomer spike which do not covers the planar end of the viron. Beneath the lipoprotein envelope is the matrix M protein layer. Nucleic acid shows a helical symmetry containing a single standard nucleic acid negative sense RNA genome and RNA dependent RNA polymerase. Animal susceptibility: all warm-blooded animals including man are susceptible to rabies infections. Man and dogs are intermediate susceptible. Pups are more susceptible than adult dogs. Street viruses: the rabies virus is isolated from the natural humans or animal infections is called as a street viruses. Such viruses produces a fatal encephalitis in laboratory animals after a long and variable incubation period of 1 to 12 weeks. The intracytoplasmic inclusions bodies, Neger bodies, can be demonstrated in the brain of these animals. Fixed virus by several serial intracerebral passages in rabbits. The virus undergoes certain changes and it is termed as a fixed virus. The fixed virus is more neurotoxic. It produces a fatal encephalitis after intracerebral inoculation and the incubation period is short and fixed to 6 to 7 days. The Neger bodies are usually not demonstrable in brain of animal dying of fixed viruses infections. The fixed virus is used in vaccine protections. Disease: rabies is natural infections of dog, foxes, cats, wolves and bats. Dogs, fox, vampire bats are important pathogen host. The man is infected by the bite of rabid dog or other animals. The incubation period varies from 1 to 3 days. Sometimes it may be short or as 10 days. Laboratory diagnosis in humans. The immunofluorescence test. The viral antigen can be detected in corneal impression, smear and facial skin biopsies or saliva by indirect immunofluorescence using the anti-rabies serum tagged with the fluorescent by isothiocyanate. The diagnosis may be made post-mortem using brain as a specimen. The use of monoclonal antibody instead of a crude antigen makes the test very specific. Demonstration of a Nagri bodies. This is demonstrated in brain but may be absent in some 20% of human cases. The Nager body appears as intracytoplasmic oval or round purplish pink 3 to 27 micrometer bodies with a characteristic basophilic inner granules. The Schiller technique, basic fusion and methylene blue in alcohol is used to demonstrate this body. Isolation of virus: the virus can be isolated from specimens like CSF, saliva and urine by intracerebral inoculations. In mice, more rapid and sensitive method is isolation of virus by tissue culture. W138 BHK21 cell lines are used. PCR, reverse transcriptase PCR, can be used for the detection of rabies virus RNA diagnosis in animals. The brain of a rabid animal is removed. The portion of the brain should include the hippocampus and cerebellum as a Neger body or abundant there. The following tests are done for the diagnosis. Immunofluorescence: this is a rapid test done on the brain tissues to demonstrate the viral antigen. Demonstration of a Neger bodies: this is still the method most commonly used. Failure to find a Neger body does not exclude the animal is free of rabies. Isolation of virus: the brain tissue suspension is injected into the young mice by intracerebral route. Prophylaxis: the pre-exposure prophylaxis. It is necessary for laboratory personnel and those who handle potential infection animals. Post-exposure prophylaxis: it involves local treatment, hyperimmune serum and vaccination. Local treatment: the wound should be thoroughly washed with soap and water. It is very important as a soap activates the virus by distracting its envelope. After washing, the wound is treated with quaternary ammonium compounds, citaveron or tincture iodine or alcohol 40 to 70%. In severe wound, the anti-rabic hyperimmune serum may be infiltrated around the wound. Damaged tissue is excised but wound is left unsutured. The anti-tetanus vaccination and antibody may be used to prevent sepsis. The biting animal should be watched if possible for 10 days. If animal remains healthy after 10 days, the rabies may be excluded. Hyperimmune serum. The human anti-rabies immunoglobulin 20 international unit per kilogram body weight is reserved for high-risk cases. Half dose of anti-serum is infiltrated locally in wound, and other half is administered by intramuscular route. Vaccines are of two main categories: neural and non-neural. Non-neural vaccines are being used increasingly because neural vaccines are associated with a serious risk of neurological complication. The fixed virus is used for preparation of all vaccines. Commonly used cell culture vaccines against rabies are shown. Manufacturing and use of neural vaccines have been discontinued in India. Vaccination scheduled. All three cell culture vaccines are available in India. HDCS, HCES, PCV vaccines have the same dose schedules. It is also same for both adults and children. Pre-exposure vaccines. There are these three doses of 1 ml on days of 3, 0, 7, 21 are given intramuscular route. The booster dose is recommended after 1 year, and then 1 year, 5 years, every 5 years. Post-exposure vaccines. 1 ml is given by intramuscular route in deltoid region on the day 0, 3, 7, 14 and 30, with the booster dose optimum on day 90. This course gives protection for at least 5 years.